Hi, I'm John Cassel at Wolfram Research. I'm going to be talking about representing biological sequences in the Wolfram language. We're going to show you how to bring in DNA, RNA, amino acid protein sequences and do all kinds of great processes on them, all your typical translation, transcription, all those kinds of things, finding features inside of biological sequences such as genes and mutations, and altogether, you're going to see that Wolfram is making great strides for processing genetic data. Hi, everybody. Thanks for coming. I'm John Cassel. I'll be talking about representing biological data in the Wolfram language. We'll start by kind of introducing what this is all about. We're really going to be introducing some near-term functionality. So under development, some things coming out in kind of undocumented or documented as experimental in 12.1 and kind of moving into 12.2, and kind of a broader functional picture of just kind of where we're thinking about taking this whole domain, and then we'll conclude. So biological sequence processing, it's analysis for discovering relative comparisons to all different species, different individuals, different cell types, different developmental stages, pathological versus, working, all of those things. And that's enabled by representing biologically similar sequences as, kind of linear sequences of text, really kind of exploiting what makes is possible through that representation. So we're sharing some proposed features here while they're still under development to elicit collaboration feedback every time we bring this stuff to new people. There's a lot of great suggestions, still a lot of things that can happen here. So really excited to hear what any of you might have to share. So just to kind of give a very brief, kind of fill in some biology background if you've wandered into this talk by accident. So here's some DNA. It's two strands together. They only bind together if the residues map in complementary pairs. And this really allows the kind of the maintenance and duplication of heritable information. So this is what allows evolution and data to be processed from generation to generation. So, but in these pairs, kind of each three of these forms a codon that's then translated further. So here we see that we have the structure of of the gene. We say here are the parts of the sequence that regulate it, that tell when the gene should be activated versus not, which determine what kind of cell it is and all that good stuff. Then once you're into the gene proper here, that's transcribed into messenger RNA that has just a little bit that allows it to feed into the part that actually can build the protein, the ribosome. And here's the part that actually does the protein, which does kind of the work of the cell. So we can, represent these sequences. So here we have, let's see here, is that probably pretty small for you all? But I guess, so we have a DNA sequence. We have kind of all its letters, its length, and its composition broken down. RNA also supported. Amino acids, similarly, we can break those down. It doesn't have to be a particular sequence. Oftentimes in the lab, we just know, it's this many terms long. We don't know what it is. That's fine. Degenerate sequences, so there's a kind of a standard alphabet that says, what bases are allowed. For example, A is one chemical, and B just happens to be everything that's not A. So we support all of the standards for DNA and RNA and amino acids as well. Oftentimes, we'll have some kind of other unsupported, thing that's just really kind of handy to represent. Like if we're adding a particular chemical to cap off a sequence of DNA, something coming off a piece of lab equipment, particular indicator. It's good just to have something that you can kind of just throw on there that's unspecified. And if it's a known type, you can convert it later if you didn't specify the type at the time. If to later determine what it is. And you can build these compound sequences. So this is really handy for the case I described before, where you have some DNA sequences coming off of equipment and they're capped with like a fluorine or something, you could just tack that on there and represent this as a mixed sequence type here. So this is integrated with our kind of our entity system. Kind of these biological sequences, they represent kind of the space that individual genes represent kind of points within. So here we ask this gene for its reference biosequence, so get back a biosequence, and it's annotated with the species and the chromosome and the orientation and positions and all kinds of good stuff like that. Similarly, if you take BioSequence and you just give it a gene or a protein, it'll give you the most appropriate biosequence for that entity. So supposing you have a biosequence and you'd like to find some information about it or some features with it, you can do that with FindBioSequenceFeatures. Here's an example where we say, here's some arbitrary sequence. We don't know what it is. We don't really have any information about it. We're going to say, where could that be on the human genome? And it says, OK, we found a biosequence position. It's on chromosome 16 at this particular position on the plus strand from this particular assembly of the human genome. And that corresponds to this particular position inside of the given sequence. So even if this sequence has some mutations or what have you, it'll still seek through and try and find, a good foothold that will match. Similarly, you can find, if you have a gene that you're looking for, you can find where that gene corresponds to. So in your biosequence along this range, that corresponds to this segment of the gene. Or if you want to find kind of just all of the human genes within your biosequence, that you can do that too. Say you're looking for maybe a particular mutation. You've read in the some journal that this particular mutation is now associated with a particular disease, and you're worried about that, or some positive trait. You can find where that corresponds to the kind of your sequence at hand. Or you can just discover all of the mutations that one might have. So this by default will return kind of the feature that's found. So it'll find the segment of the gene in this case, and so the feature and the position along the biosequence that you gave it. If you'd like to extract the sequence associated with the found feature, you can do that by just applying this sequence command here. And this sequence is your version of the sequence for that gene. So this is, we have some support with molecule. The chemistry developers will be exposing this sequence molecule here eventually. And with that, we can give them biosequences and translate them into the underlying molecule. And then all of the chemistry stuff will also apply. And not only, we also have some functionality that applies to, we're extending some of our other existing functionality, random instances of biosequences. So you get 3 DNA sequences of length 50. So one kind of neat feature is if you take random instance of these degenerate sequences. So S can either stand for G or C. So you can kind of describe the space that you want to kind of sample within using the degenerate terminology. You can also use weighted choices. So here's sampling from A or C. And you can also use degenerate terms in this sampling format. So this is 20% will be A, and then 80% will be everything that's not A. So 6 sequences, each 9 long. Sequence alignment also works on biosequences, thank goodness. But also it has some knowledge of these degenerate bases so that GG can match NN and NT can match, so that you actually can get like, a kind of a perfect match between different degenerate sequences if they're supported. Similarity.
And if you have an uncalled base or something like that, you can put a wild card in or so.
Yeah, N is really kind of the wild card. It's in any base. So, and that'll match. Similarly with sequence similarity, here we say that by allowing these similar degenerate bases, these sequences are more similar. And Needleman-Wunsch also supports this improvement. Longest common sequence positions, subsequence, this entire family also works, also matches degenerate terms, so that you can find, matching subsequences here and have all that respected. So now kind of moving on to complements and conversions. So earlier we saw that the kind of the DNA strand was two-sided. So when you look at kind of the other side, you get the complement. So here AGG translated to TCC. Often, biologically, they're often actually read in the reverse order. So we also provide kind of this reverse complement here. Transcribe converts the DNA into RNA, and inverse transcribe will convert it back. Translation, so this is the process by which it's actually converted into the protein, into the sequence of amino acids. So here we see the one translation and an inverse translation to take it back. Inverse translation can actually, many different codons all correspond to a particular amino acid. So this one is by default you get a sample, but if you supply all, you can get all combinations thereof. There's a BioSequence inverse translation count, so you know how many of them you're getting. Actually, I'm just going to go off script here. So that blows up really fast, so that's a good thing to have. Translate, we support, entities for all of the common sequence translation tables supported by standards. So if you have mitochondrial DNA or some weird archaeal DNA or so forth, you can have a translation that works for you. Oftentimes, you're going to end up in the situation where you're not kind of perfectly aligned, where you have this kind of frame shift sort of phenomenon. So because every 3 letters is what's translated, if you get off by 1, that's a big problem. And it really kind of messes with sort of your analysis there. So we offer the ability to just jump to a start codon so that you end up with kind of the translation that's appropriate. Or if you'd like to manually adjust it, then that's provided to move either forward or backward on the frame, either dropping terms or adding degenerate terms as necessary. So that's kind of a lot to keep track of. And if you'd just like to just convert it for me and I don't care about the details, then BioSequence Convert is your friend. You just go ahead and give it the type, and it will convert it to whichever one is most appropriate. Again, all here will give you all the cases. And there's a BioSequence conversion count, so it doesn't blow up in your face. So I guess we also are supplying some functionality for matching and validating biosequences. So is this actually a legitimate biosequence? Are its terms fully specified? Is it DNA? Is it fully specified DNA? All of that. And we also have a biosequence match queue to handle kind of matching these degenerate terms. So some more degenerate term resolution here. If you have something where you have a degenerate term and you'd like a sample that's some fully resolved sequence, well, that can be provided. Again, the same convention. You give it all, you get back all the combinations.
So resolve degenerate, is it like a random instance of something that would suffice? Or?
Yes and no. So it's guaranteed to be the same each time, but it is arbitrary. So if you run it again and again, you'll get back the same.
Okay.
There's going to be no surprises in terms of it's not a random instance, but so it kind of gives you a choice there. And same story, BioSequence resolution count avoids a combinatorial explosion. So that's some of the functionality we're going to be releasing in the near term. I guess we're really looking at quite a lot of different functional areas to extend this to. I've highlighted some here that I'll be looking at in the next few slides, but certainly if you see anything of here that's interest, please let me know. So one common problem is that you'll be interested in having built-in support for a type that we haven't provided, such as if you have some archaeal transfer RNA, or if you have Hajimochi DNA, or you have some new synthetic alphabet that you'd like supported as though it's first class. I think we're really interested in that, in using kind of the entity store mechanism to allow users to provide their own types that can then convert between our types, such as, for example, making some organism-specific wobble complements between our RNA and some ribosome-specific tRNA definitions. We're also really kind of interested in some data indexing and providing search and feature annotation capabilities. I mean, right now we have some genome lookup, some SNP finding that you've seen, and we potentially do protein lookup and others as there's demand for them. One thing that seems to be a relatively short follow-on extension from this is kind of doing the 23andMe thing, labeling these SNPs by what features they correspond to, what diseases they correspond to, heritage and evolutionary markers to, so give me all of the SNPs in this gene that correspond to some set of traits or some set of diseases. Could be very interesting. A lot of times, we're going to be in the situation where users are looking into they're researchers, they're really looking into their particular species, or they're really looking into something that's proprietary. And so be able to use things like FindBioSequenceFeatures for their data is kind of taking the index stuff that we have built internally and being able to expose it is of interest. And also there are so many great bioinformatics resources out there to be able to then provide an API, not worry about building a separate index, to use something that you've already built is going to be key for us, I think. So we're also looking at improvements to alignments and structural comparisons. The many-to-many feature-to-sequence alignment is very popular, doing RNA sequencing, taking a tabulate biosequence features where we have kind of many different sequences coming in, and then kind of putting them together. That's closely related to sequence assembly, both where you have some reference that you're going against and you're building kind of the assembly on the fly without reference. We're looking into expanding our sequence alignment from pairwise to multiple alignment, kind of some many-to-many alignment comparisons that go with kind of the RNA-seq school before, the really differential expression, seeing when, if normal versus disease situation, really being able to display and compare those and determine if those are statistically significant for a given pathway with enrichment analysis. So I guess the other big problem or challenge that we're facing is that biological sequence processing there are so many scripts and tools out there that are really kind of distributed, and it's deployed in pretty much every single possible way. Containerized deployment is becoming progressively more popular. And we really think that kind of Wolfram notebooks and the Wolfram engine is really kind of fits really well into this, both as kind of like a prototyping platform for then deploying containerized scripts, and then also as an environment for receiving the results of containerized workflow. So in conclusion, we're really starting to feel how Wolfram Language can support biological sequence processing. And there's many directions, as you've seen, in which this work will develop. And so your suggestions can still be incorporated into our designs. Thank you.